Hello, everyone. Welcome to the GEMS podcast. I am so glad you all are here.
Hello, everyone. Today we are so lucky to be joined by Jazmine Benjamin, a Ph.D. student at the University of Alabama Birmingham. Jazmine, thank you for joining us. Could you tell us a little bit about yourself?
Sure. So I am just starting my fourth year of a PhD in cell molecular and developmental biology at UAB, University of Alabama at Birmingham. My research focuses on the role of feeding behaviors in diabetic nephropathy, or put a little bit simpler, how what you eat and what time of day you eat can lead to diabetes and kidney failure as a result of that. I have been involved in science for probably about 7 or so years now, so kind of halfway through undergrad. I really got interested in science and kind of took off from there. After I graduate, my plan is actually to go into policy. So working on Capitol Hill or working with lawmakers to kind of advise on policy as it relates to STEM-related topics.
Awesome. So is there an experience in your undergrad journey that inspired you to go towards STEM, or was it before then?
So I think it's really interesting because even when I was a child, I was very interested in kind of STEM things. So I was always outside, kind of running around and poking into things. I had Legos, I had magnetics, I had Hot Wheels. My fourth grade science project was on centripetal force. So I've always kind of really been interested in science, but having parents who aren't necessarily bench-top scientists, they probably weren't able to really identify that with me. And, of course, I also didn't have necessarily a scientific role model to look up to. And so when I was in elementary school, a lot of my textbooks did not have people that looked like me. So I really didn't think that I had a place in any kind of STEM field. And so I really lost my interest in STEM until my sophomore year of undergrad. I was taking kind of our introductory cell and molecular biology course, and Dr. Hancock was our professor, our instructor for that course. And so in the lab portions of that course, I would always just kind of pester him with questions and about everything. You know, is the difference between corn and maize the same thing as the difference between a plantain and a banana? And, you know, questions like that. And so towards the end of the year, I ended up getting an A in the class, and you know, he told me, you know, you should switch your major from, I think it was chemistry at the time, to biology. And I was like, you know, I want to go to medical school, you know, and he's like, well, there are things outside of medical school that you can do when you're a chemistry or a biology major. And so he kind of took me under his wing, and he became my academic advisor, and he gave me a spot in his lab, and he really kind of helped me to realize my kind of strength in being able to conduct research, but also talk about my research, which is really important in terms of grad school. And he also kind of opened my eyes to the fact that I didn't have to be a doctor. I could still be a scientist. I could still do research and do the things that make me happy. So I kind of attribute my start in science partially to my parents for, you know, always making sure I had little toys and things to keep me busy, but also definitely to Dr. Hancock for kind of noticing that I still had that curiosity and helping me to turn it into what is hopefully going to be a great career.
So as you were at Dr. Hancock's lab, is there a specific research project that you participated in that you'd want to share about? Is it the
Sure.
the MPing? Is that correct?
Yes.
Okay.
Yeah. So MPing, kind of like my first scientific love. So it is our lab, very simply put, we kind of looked at genetically modified organisms, which is always kind of a hot topic. But we were essentially looking at MPing, which is it's a transposable element. So essentially it has the ability to kind of jump from one gene to another and affect how our plants look. So if you've ever seen, you know, a rose that maybe is like part white and part red, or an ear of corn that has kernels that are all different colors, that's due to transposable elements like MPing. And so my project was really looking at the behavior of MPing and how we can kind of make small changes in its DNA in the DNA bases and important parts of imping to kind of figure out what exactly makes it jump more or less. And so this could be really helpful for if we're trying to develop, a drought-resistant strain of rice, or perhaps plants that have bigger leaves that can protect the actual crop underneath better from the sun, things like that. So kind of just trying to tackle food crises by looking into this element that's in a lot of our corn and rice and kind of trying to figure out exactly what makes it work.
So did that research have an effect on how you decided what your PhD research would be about?
Yes, I think that the experience itself really helped. So I learned that I really love doing things with my hands and working in kind of small teams and talking about my research. But I also, thanks to Dr. Hancock, had the opportunity to do a summer research experience at the University of South Carolina in Columbia, where I worked with Dr. Jason Stewart, and it was much more cell molecular biology in the context of humans. And so we were looking at this particular gene that's involved in kind of keeping DNA replication going. And so that's really important in terms of cancer, where sometimes you want replication to continue and sometimes you don't. And so kind of in combining those two experiences helped me to see, A, that I really love doing research. I love talking about my research and everything, but also that I love doing research that kind of contributes to the greater good, but I really love doing research that was kind of more biomedical leaning.
So I was researching just a couple of things that you were a part of, and what is the STN1 knockdown on DNA replication? If you could just explain it in more simpler terms for me.
Yeah, so that was the summer experience I did with Dr. Jason Stewart. And so I don't really know the science background of the audience for this topic, but essentially in cells, we at certain points replicate the DNA that's in our cells. And so this is kind of mitosis, essentially. And so when that happens, we have these helicases that will unzip our DNA and allow those bases to be replicated so that we can turn one strand of DNA into two. And so the CST, which is CTC1, STN1, TIN1, that complex is a really big part of the replication fork. So this is the actual area where the DNA is being unzipped and those bases are being rematched to create two strands of DNA from one. And so what can happen, especially in cancers, is that this replication fork can become unstable, and essentially it will stall or it'll stop replicating. And if it stays stalled for too long, it'll completely fall apart. And so that's when you'll end up with DNA that's not completely replicated, which can give rise to various kinds of cancer. And so this STN1 part of this complex, obviously all three parts are very important, but for the context of my project and the time that I was working with Dr. Stewart, I focused on the STN1 part of the complex and kind of how important is that particular piece of that complex. If we take STN1 out of the equation, or if we knock it down, we decrease the levels of STN1, does the replication fork still stall as much? Does it fall apart as often? Is replication better? So that was kind of the premise of my experiment, is kind of, let's take out this one block and see if the tower still stands or if it falls over.
Great. So, hold on. Okay. So I also saw that you did a interactome. I don't know if I'm pronouncing that correctly, but
yes.
It's a map, essentially, right, of molecular interactions of a cell.
Yeah.
So what type of cell, like, what was your interactome of, if that makes sense?
Mm-hmm. So in that experience, I was looking at essentially inside of cells, you have obviously proteins that move from, place to place. So from, maybe the endoplasmic reticulum to the Golgi apparatus, or the Golgi to the cell membrane. But you have to have kind of vesicles that enclose your cargo and protect it from the cytoplasm as it goes so it doesn't get degraded or anything. And so I was looking at what exactly makes those vesicles kind of work, what makes the vesicle kind of pull off from, say, the endoplasmic reticulum and fuse with the Golgi apparatus. And so I was looking at this very specific molecular switch that's involved in that process. And so the interactome that I was working on is trying to figure out what is the cue that turns that molecular switch on so that we can move cargo from the endoplasmic reticulum to the Golgi apparatus. And so the interactome that I created was essentially this one molecular switch. What is every gene that anyone has ever seen in an experiment that it could possibly interact with? So it's very abstract. And essentially that's what an interactome is. It's kind of, you pick a point that you put in the middle, and then the software that I use essentially looked through all of the literature that, is widely available and found mentions where someone said, hey, this gene that you're very interested in, this molecular switch, may interact with this, or we've seen it interact with this. And then I was able to use that interactome to kind of narrow down the pieces that might be part of the puzzle to kind of making this molecular switch work. So the interactome was kind of the beginning of that project.
Wow. So as you have been working in the lab and as a student, have you seen an apparent gap between male and female roles, or have you faced any type of adversity in that kind of realm?
Let's see, I'm trying to think. So I think that there is this kind of this gap between male and female roles, but I think that my university, at least the department that I worked in, was actually pretty good. So our department was majoritively women in terms of people who were running labs. Now, obviously, our department chair was a male, and I think that most of the department chairs in the kind of scientific side of our campus are male. So that's obviously, something that I'd like to see more women kind of take on those roles. But I think as far as being a student, I personally don't have really any experiences where I've directly seen myself treated differently from a male colleague. There's obviously also always, in classes, guys who, kind of want to puff their chest out and show that they know everything. So, they'll be a little more boisterous about answering questions and kind of wanting to argue with you about questions and things like that. So that's always a factor. But in terms of just competition or kind of working together in a lab space, I don't really think I've seen much of a difference in terms of gender, fortunately.
So that's amazing. And going throughout your day, what does that look like as a PhD student?
So kind of like what's a day in my life is your question?
Yes, a day in your life.
So that's kind of one of my favorite things about being a PhD student is that no 2 days are the same. At least before, a pandemic, no 2 days were the same. I would say, I guess I'll give you two parts. So earlier this year, before, everything kind of shut down, I would have days where I was really busy. So I had a lot of experiments to do and it was a lot of running around and I, barely had time to eat. So I would come in, set myself up for an experiment, maybe check my email while I'm waiting for things to thaw out. And then, the rest of my day is spent running that experiment. Every now and then we'd have kind of seminars to go to where we could listen to someone else talk about their research. So I might take a break from my experiment to go to one of those, but then it's right back to doing my experiments. So some days are like that. Other days are, I come in, I, of course, check my email like I do every morning, and then I'm just reading papers and I'm writing, kind of either summaries of the papers or I'm working on a grant or I'm, reading, a draft of someone's paper or something like that. So there are days when, I was always on my feet and then days where I was just kind of sitting around all day. But right now, kind of working mostly from home, a lot of my time at home is spent reading papers, working on grants or, reading drafts of other people's work, and then also doing a lot of my experiment planning and my data analysis at home, and then only really going to the lab when I need to check on an experiment. So right now I have some mice that I'm working with. So if I need to check on my mice, then I'll go to the lab for that. So yeah, it's kind of doing actual lab work in the lab, and then everything else is being done at home.
And with those mice, you're studying nephrology, correct? So with the kidneys and diabetes.
Yes, yeah. So right now my research focus is the role of feeding behaviors on diabetic nephropathy, or essentially what we eat and what time of day we eat can contribute to diabetes and the kidney failure that can be a result of diabetes. And I'm using mice for that.
And what was the process in selecting that type of research? Was it what the person in the lab was doing, or did you find a gap in other people's research and that was what made you want to pursue that type of research?
So I guess a little of both. So it was really more of that's what my boss was doing. That's what he has money to research right now. And I also personally think it's very important to research. Obviously, we're in a culture where, obesity rates are going up. So of course diabetes rates are going up, and, kidney damage is a result of diabetes and high blood pressure and all those things that come with being obese. And for me, being in the Southeast where we have, some of the highest rates of obesity, this research is extremely important. And that's likely why my boss gets so much money for it. But I really got into the research topic. I knew I wanted to stick with diabetes. And my boss had, a good bit of money to kind of pursue this question, but I also thought that it was really important. So it's kind of, there's this gap in knowledge that we really need to address, but also, my boss has the funding and has garnered the interest for the topic. So I just kind of stuck with it.
Absolutely. Is there any type of advice that you would have for high school girls that are looking to pursue STEM, or just anyone in general who's looking to gain a PhD as you are?
For high school girls, obviously they have to go through undergrad first. I would say the best thing to do is to definitely find yourself a mentor or a network of, people who have kind of been there before. I think that for women in STEM, there are quite a few networks where, people of all levels, undergrad, high school students, undergrad students, grad students, faculty members, that they all kind of meet up and you can kind of find a mentor that way. But I think that the best thing to do would be to find a mentor who's in the place where you would like to be, that is committed to kind of helping you along the way, because it's really not easy. And then for those that want to get a PhD, I would say, really get that research experience, as much of it as you can, in as many fields as you can, especially if you're not entirely sure about what it is that you want to research. But learn some new techniques, become familiar with the vocabulary and the jargon that's used in science, get comfortable talking about your science, find yourself a good mentor, again, who's doing research, whether it's in something that you want to spend the rest of your life researching or not. And just don't give up. PhDs aren't easy. They're not meant to be easy. That's why a very small percentage of people in the world have one. But it's very important that you find mentors and you find a support system and a network that's willing to kind of help you through the tough parts.
Thank you so much. And I wish you luck in the final steps of gaining your PhD.
Thank you.
But that's all of the questions that I have. On behalf of all GEMS, thank you so much for letting me interview you today. Thank
you so much for giving me the opportunity.
Well, thank you so much again. I didn't
You're
take the full
welcome.
hour, but I just, thank you so much for just giving your time.
Of course, you're very welcome.
Thank you all for listening. If you have any questions or comments, you can contact us through Instagram, Twitter, Facebook, at KolgaGems. You can also reach us through email. Our email is kolgagems@gmail.com. That is COLGAGEMS. And I'll see y'all next time.